Hey guys.
Thank you.
Okay, we're only 20 votes apart now.
Excellent. Excellent.
It's 236 to 256.
Yes, so please vote for Jack the dog. Now, section 22.3. This is one section to round out chapter 22. It's kind of a shorter chapter. This is focused on these four major areas where we obtain evidence to support the idea that life changes on Earth through natural selection driving evolution. Life changes based on evolution through natural selection. And I remember what I was going to say. You have all these slides, so please do not feel like you have to write every single thing down. And what we're going to do today is look at these four arenas of the world where we do most of our time researching and finding evidence to support evolutionary thinking. So, this first one, through direct observations of change over time in populations. Your book has a few examples, but I was going to talk to you about what we call MRSA. MRSA. This is methicillin-resistant Staphylococcus aureus, sometimes known as staph. You probably have heard of like staph infections.
Yeah.
A certain proportion of the population, they say about 1 in 3 people, carry staph kind of as part of their natural bacterial population on their skin and around the nose as well. If you have had, or if you will one day take, Miss King's anatomy class, they actually will swab. There's a lab they do where they swab your skin and see if they can grow this bacterium and see if you're a carrier for it, basically.
We did that last year.
And you did that last year too? Okay, good. So what we discovered with this Staphylococcus aureus way back in like the 1940s is that it has a knack for developing a resistance to different antibiotics that we use to treat infections, including infections that are caused by this bacterium. Now, again, MRSA is specifically this methicillin-resistant bacterium. And if you've ever heard of like kind of the flesh-eating infections, it can be caused by a strain that has a very unusual name. It's called Staphylococcus aureus clone USA 300. I have no idea where that name came from. But that's a strain of this resistant bacterium. And I'll tie it into evolution here in a second. This is actually the plasmid, the ring of DNA that this prokaryotic organism carries, and they have mapped out different regions associated with like its ability to live on your skin, how severe an infection can be, and its resistance to this antibiotic known as methicillin. We know where these genes are on its genetic code, on its genome, on its plasmid. And again, these are genes that allow it to do certain things. Now, what's more important is really the timeline here that you see at the top, where in 1943, we began using penicillin to kill Staphylococcus aureus when it was causing an infection. And in only 2 years, the world population of Staphylococcus aureus in response to this environmental change, in response to penicillin showing up in its environment, had already had part of its population branch out, and you started seeing cases of penicillin resistance. So what do we do when a bacterium, when a germ is resistant to an antibiotic? We make a new one. Yeah, we make a new one. So we introduced methicillin in 1959 as a new weapon to fight this living thing, this prokaryote. And within 2 years, we started seeing strains, the population, parts of the population had changed and adapted to this new environment with methicillin in it. Which its environment, a lot of times, is us. And the antibiotic was that we were taking was a change to the environment. Now, there's a few key points I want to make, and there's a lot written on this next slide that you should kind of just read through later. I don't think it's anything too complex. It talks about how a lot of these antibiotics like methicillin works, which is not really that closely connected to this concept of evolution, really, but it is in a way, because methicillin basically can kill some bacteria by shutting down an enzyme that the bacteria needs. It needs it to build cell walls, which is an important part of the structure of these cells. But what does the resistant strain do? It's a mutant that just makes and uses another enzyme to do this. Well, that's a genetic change, which we call a mutation, that crops up, causes the individual to have a different trait, which is resistance to this chemical called methicillin. And that trait is an advantage and begins to show up more and more and more because the individuals with that trait are surviving and reproducing. Now, how does a prokaryote reproduce? Say again. Binary fission. They just split from one cell into two. So this is asexual reproduction. It happens very quickly, and I think somewhere it mentions that natural selection can occur rapidly when you have short generations. So if you want to see a population changing over time, study why it's changing. How does it adapt? What are the adaptations? Where do they come from? Why are they successful? Bacteria are great for studying this stuff because while it might take a human 20, 30, 40 years to have a child and reproduce and create the next generation, bacteria can do it in an hour. That's a much faster rate of reproduction. And that's, just their strategy for surviving on this planet. Just keep dividing and make as many of you as you can. We have a different strategy. Now, another point I want to point out is that this process of natural selection, selecting survivors, having them reproduce, and then passing on their genes to future populations, natural selection does not make the new trait. Okay? Evolution, when we talk about evolution and the change of life, that process isn't the process that's creating new traits and creating variation. It's mutations that do that. All right? So we've talked about mutations. Mutations are changes in DNA which might be harmful or fatal or neutral or actually beneficial. And when they're beneficial and they help you, then that mutation can start to spread throughout a population as time passes and you start seeing changes as a result. So evolution through natural selection is simply this concept that based on your traits, you may be more or less successful in your environment, and you might have more or less children, and the future may look more or less like you. It's mutation that actually gives us these new traits. All right. The next arena that we pull data from when we study evolution through natural selection is this concept of homology. Now again, there's a lot written up here. I try not to use slides with a lot of writing, but you have these slides written. So just use them later for reference. Homology, what does the prefix or root homo mean? Same. Just like homogeneous is same throughout. Homology is basically where we look at the things that make us similar between, say, two different species. So when we talk about homology in general, we're talking about similarities that one group has with another group that are the result of them being related. And what we really look at a lot is what we call homologous structures, which homologous clearly comes from this word homology. Homologous structures. Actual physical structures that you have, a lot of times it's like bone and teeth that we're looking at, or layers of tissue. But these are structures that are similar between groups because they come from an ancient population where they were the same. So an ancient population splits into two groups. Those two groups should have similarities, and we study these similarities. And honestly, for me, this is one of the most powerful, personally, one of the most powerful pieces of evidence that I've seen for this idea of supporting this idea of evolution through natural selection and the idea of the tree of life, kind of the relatedness of all living things. And so here's a diagram that I think is used quite a bit. This is the forelimb, the front limb, which in humans is the arm, of 4 different mammals. So these are all mammals up here. Now, if you don't look too long at this, and maybe just close your eyes and think to yourself, my arm, a cat or dog paw and leg, front leg, a whale flipper, and a bat wing. You probably are thinking, man, those are really different structures. But they're actually homologous. They have a lot of similarities because we are actually all distantly related if you go back like 60 million years on the family tree of everything. So what you see here is a color coding of our humerus, radius, and ulna, which, by the way, the flapper chicken wings that you get, you have the drumsticks and then the drummies and flappers. That's the radius and ulna, those 2 bones of that bird. So they have them too. But our humerus, radius, and ulna, and then kind of the hand and finger bones that are these carpals, metacarpals, and phalanges. So you can see in the human arm, this is probably fairly familiar. You can see some of these bones, feel some of these bones in your own arm. And if you look at the front leg of a cat or dog, you see the same general arrangement of the same bones. Now they have clearly been modified. They've clearly changed over time. So I'm not saying that they are the exact same bones or the exact same structures, but they are the same bones that form in the same way from birth to adulthood, form from the same tissues and cells, and they're in the same general arrangement. But what really, to me, drives home this idea of the relationship we have to other living things is the whale flipper. And you don't typically see the bones of a whale flipper. You may have seen a whale before. You may have seen whale flippers like splashing on the water on TV or whatever. But look at the bone structure. The whale flipper has the humerus, radius, ulna, hand, and finger bones. It has finger bones inside the flipper. Why would you make a flipper with finger bones? You just wouldn't. All right? So there are the finger bones that are greatly modified, but still exist in that same general pattern. And then the bat wing as well. I mean, the wings of a bat are just the arms with like this membranous skin. And you can see here that it's the hand and fingers that make up a major part of the wing of the bat itself. Homology. You wouldn't even think these are the same kinds of structures. But when you start looking at like bones inside, they are clearly based on the same general patterns. We also compare things like embryonic development. Now, they don't really look a lot alike, this embryo of a chick inside an egg and this human embryo inside the uterus. But you do have structures like these pharyngeal arches that in humans close up during development before birth. Same with chickens. They don't close in fish embryos. What are they in fish embryos?
Gills.
They're the gills. Now, not the oxygen extracting parts of the gills, but the gill openings, the kind of physical structures. So the fact that we have structures like this and other organisms have structures like this, even in the embryonic stage, as well as a tail, is evidence of a relationship of common ancestry. There's also some weird stuff like vestigial structures. These are structures, and it's probably a fairly technical definition. Vestigial structures are essentially, when you hear the word vestigial structure, just think like no longer used. Basically a useless structure. Which is also kind of a weird thing to have. Can you tell me a vestigial structure in a human? Sometimes it gets infected, swells up, is very painful, and they have to take it out. The appendix.
Would the
Yeah.
gallbladder be, I mean, I know you can remove gallbladder, I don't know.
The gallbladder is interesting because you can have it removed, but it does help you it helps deliver chemicals to your intestines that help you digest like fatty foods. So when you have your gallbladder taken out, sometimes you kind of have to watch your diet a little bit, or your stomach might hurt. So I don't want to cast it off as useless. But your appendix is basically like a cecum, which in a lot of mammals is used sort of like a second stomach, where extra digestion can occur if you're eating a lot of, say, grass or...
I heard that's where like a lot of like bones go sometimes. When you eat like, sometimes if you eat like, if you swallow fish bones by mistake.
Oh, like a more recalcitrant, harder to digest thing. Things can get stuck in your appendix, and you can get it infected. And the problem is that it's no longer like a stomach-like sac like you might see in other animals. For us, it's kind of like just a little sac like the size of your pinky just hanging off of your intestines. And the problem is that, junk just kind of can slip in there and it can get infected. And really, that's the only thing the appendix seems to do for us is send us to the hospital for emergency surgery. That is not a useful organ, but the fact that we have it and other organisms have it and use theirs does seem to indicate a relationship. You can see this in whales. We're going to bring up whales again here in a minute. This is actually a different slide. This fits along with homology here. We have a lot of, major groups of animals in the modern day. And based on some of these structures and who has them and who doesn't have them, we can start to map out major groups and then refine that down as we get other bits of information. So these numbers are like major branching points where everything that goes back to a certain number was actually kind of one population at a time, and that population had portions that split off from each other and changed over time since then. And then you have these hatch marks that indicate what common or homologous structure all of these share that cause them to be different than something else. So here, like all of these organisms, and I mean, if you went back on the family tree to where we branched off from amphibians, you're talking 300, 350 million years ago. So we're going way back here. This isn't like thousands of years ago, we were all amphibians. I'm not saying that. But once you get a living thing that has a limb with digits, like fingers or toes, we can go ahead and start separating all of those things from, say, the lungfish that doesn't have limbs with digits. And then as animals moved on to land, it was very important to have basically a sac around the developing embryo, which you have in the eggs of birds and in the uterus of mammals, or in the eggs of mammals if they are egg layers. Anybody know an egg-laying mammal? The duck-billed platypus lays eggs. What? It's a mammal that lays eggs? Yep. But they have an amnion. So that separates us, say, from amphibians that don't need to protect their eggs from drying out because they lay them in the water. And then you get down to things like feathers. Well, feathers certainly separate more specific groups from some of these other ones. So based on who has certain homologous structures, we can start branching out and creating a tree of life.
I thought platypuses were green because of Phineas and Ferb.
Oh, I do not. I don't know the show Phineas and Ferb very well. I'm old, guys. I'm old. Now, this gets tricky. And this is where Darwin made some mistakes. You have terms like pachyderm, which refers to like thick-skinned animals. And I can't think of the exact ones, but I think it's like the elephant and the rhino and the hippopotamus. Back in Darwin's time, they kind of used this idea of homology to try to put them in a group called the pachyderms. Well, we now know that thick skin isn't a trait that necessarily means you're related to another thick-skinned organism. Sometimes, and this is what we call convergent evolution, or sometimes analogous structures instead of homologous structures. But analogous structures or convergent evolution is when you get the same result kind of by chance, but not because of a close relationship. And I'll give you an example. Bats fly, birds fly, and insects fly. Well, if you look at a bat wing and a bird wing and especially an insect wing, insect wings are totally different. There is not a similarity between a butterfly wing and a bat wing, except for the fact that they are used to fly. That is a case of convergent evolution. Is flying useful?
Yes.
Yeah, it can be very, very useful. And because it's so useful, you can find in nature many organisms that just develop flight independently from one another. The eye has actually done that. There are different organisms on Earth that have eyes that evolved completely independently from one another. So you have to be careful about saying, well, those two things look a lot alike, so they're probably very closely related in evolutionary terms. Sometimes they're not. You have to take all the evidence that you can get to make some of these determinations, and sometimes we still make mistakes and rewrite family trees. Okay, the fossil record. The fossil record tells a story. We sort of talked about this already to some extent last class, but it tells a story of change on Earth in different populations. And it's also how we can see, maybe not perfectly, I mean, some fossils are in pretty poor shape, but we can actually see some of these homologous structures even though the animal isn't around anymore, or the plant.
Have all fossils been found?
No. No. We don't know how many we haven't found. It's kind of a guessing game. But there are still lots of fossil discoveries being made.
How much of the ocean have you discovered? Is it like a super low percentage?
I don't know about, like, in terms of things that live in the ocean, I mean, it just kind of depends. The ocean is still kind of an unknown frontier to some extent, but not near as unknown as it was, say, 100 years ago. We know a whole lot more now. Now, here is a chart that we have put together based on evidence from the fossil record and from modern living things. And this is a look at where the cetaceans came from, which is basically whales and other aquatic mammals. And here we have like a living whale today. But we have fossil evidence of other now extinct organisms that have, in the bones we have recovered from the fossil record, have a lot of homology to modern whales. We have this Dorniodon, which, by the way, what does the dagger mean?
Extinct.
It means it's extinct. They actually had some leg bones that seem to have protruded from the body. Some little tiny leg bones sticking out the back, or little useless legs sticking out the back. And if you go, and that's somewhere around like 30 to 40 million years ago. If you go back further, this Rhodocetus and Pakicetus, these are legged organisms. So Pakicetus is something like 45 million years ago, extinct, not in the direct lineage of whales, but a cousin of whales. They have a lot of whale homology, these Pakicetus and Rhodocetus. We can see a lot of similarities in what bones we do have. And we'll look at some of those bones here in a second. And if you look further at the homology, all whales, ancient, extinct ones, and modern living ones, seem to be related to kind of your two-hoof animals like pigs and deer.
What if there like water animals that are claimed extinct are actually not extinct and just at like the bottom of the ocean?
Well, it's funny you say that. The coelacanth is a fish that we found in the fossil record. It was from rock that was like, would be as old as 200 or 300 million years. And we thought it was just some dinosaur fish, some ancient thing that went extinct. And then in the 1930s, a paleontologist went to this like fish market, I think on the western coast of Africa somewhere, and there was a live, like a coelacanth for sale that a fisherman was just selling. And he was like, this is an extinct fish that we thought hasn't been around for millions of years. And then they found them. The coelacanth is a fascinating story. So there are instances of, kind of marine organisms that we thought were extinct but weren't. And I just saw in the news the other day, there's a bird that we had not seen for like 50 or 100 years. And somebody saw it like this month. So we probably chalked it up as extinct, but it wasn't. It just wasn't found by us. Now, some of these things, it's just, we know the globe pretty well. And we find new species every day, especially in rainforest. The Amazon is an amazing, amazing place where basically, if you just go and look, there will be a species of something that you find that hasn't been discovered yet. There's just absolute amazing diversity there. But when it comes to these, what we think were swamp-dwelling mammals that kind of had a lot of crocodilian behavior to them, we've just never seen one. There's really no good reason to think that they're still around. Your book shows some of these whale and whale ancestor living in extinct bones. And this is the nice thing about the fossil record. Pakicetus, we don't think it's been alive for the last 50 million years, 40 or 50 million years. But we can still find its ankle bone in the fossil record. And you can still compare it to pig and deer bones. And you find that it has these kind of two rounded ends on this one ankle bone. Whereas most mammals only kind of have one, like this ankle bone from this dog. So it's similarities like this that we rely on a lot. And I'm going to be honest with you. Sometimes it's just an ankle bone, and that's all we got. But sometimes there's a whole lot more information there. And you might also be thinking to yourself, well, are any of you bone experts? I'm not. I'm not a bone doctor. What do you call them? Osteopath. Yeah, I'm not. This isn't my area of expertise. I mean, heck, you could have told me these were teeth, and I'd be like, okay, I don't know. But here's the deal: if you take this ancient animal's ankle bone and don't tell them where it's from, and hand it to an expert in the field of bones, they will look at it and be like, this is either pig or deer. And you go, well, actually, it's from remains of a Pakicetus that has a lot of whale characteristics outside of this ankle bone. Even its nostril was kind of towards the top of its head, as if the blowhole was already moving to its modern location. So experts will look at these, and to them it's as clear as anything you're familiar with. It's just sometimes the familiarity isn't there for us. And then finally, last slide. I know this has been a bit of a long chit-chat here. We have what we call biogeography. Now, this is an area of evidence for evolution that is important to understand, I'm not sure there are too many gotcha moments. Like to me, it's a lot easier to be like, why would a whale have finger bones if it wasn't related to, organisms that have digits like fingers? Here, biogeography is more of a way to explain some of the weirdness that people find. And in this diagram, you have two regions filled in here. There's this region down here, the very southern tip of South America, and then Tasmania. And in these two regions, there are these two species of freshwater fish. They're two different species, but they are very closely related. All right? So, and they have the scientific name here. I'm not going to try to pronounce either of these, but these are the scientific names for these two freshwater. They cannot survive in salt water, freshwater species of fish. And all the evidence we have tells us that these two are pretty closely related to one another on the tree of life. And what people say is like, well, then, this is a problem for evolution through natural selection. How do these related species, they can't swim halfway across the world through the ocean. They would never be able to make that. These are freshwater fish, they could not get there. But what we have found through geology is that these land masses were actually connected at one point. The continents are moving through plate tectonics and what we call continental drift. So once you understand that the world has been changing, where the continents have sat have been changing, and that these regions were closely connected at one time in the past, then you start to be able to explain some of these unusual findings. We see that a lot with plants that you find in South America and in Africa that are very closely related, and you're like, well, how did the seeds get across the Atlantic Ocean? Give me a break, that's not possible. Well, it's because these two continents were once stuck together not that long ago in the grand scheme of things. Okay, next class we're going to look at sedimentary rock and try to estimate its age based on the data we get. Don't forget that on May the 2nd, Sunday, May the 2nd, by midnight, I want your video project that I will show you a sample of here in a second, and that's also online. And make sure you're kept up with your reading. We will do the lab that's posted for today, next class, but it won't be due till the end of next week. So we're going to stop the video.
Bye, guys.